So let's get started with the first presentation on human milk oligosaccharides. So what are human milk oligosaccharides and what do they actually do? That's going to be the focus of this first talk. To start off the presentation, let's really take this statement here from the 2016 Lancet breastfeeding series, where it says that the death of 823,000 children and 20,000 mothers each year could be averted through universal breastfeeding, along with economic savings of $300 billion US. And really, we are asking the question, why is that? What is in human milk that saves lives? And how does this all work? To understand that and to tackle those questions, we have to understand that human milk does not stand in isolation. Human milk is part of a co-adapting triad of the mother, of the infant, including the milk, and being embedded in an environment that rapidly changes. So human milk changes, the triad changes and adapts to the environment. Now, if we wanted to reduce it just to human milk, you see on this graph here, which you're not supposed to see, at least not in detail, that there is so many different components of human milk, and each individual one has a function. And of course, in context and in concert together, they drive infant outcomes as well as maternal outcomes. So very complex topic that we need to reduce down to individual components to really understand what's going on. So let's just focus on the macronutrients of human milk and forget about all the micronutrients and all the other bioactives that we will talk about at the very end of this session. So in human milk, we have protein, fat, the milk sugar, lactose, and then we have oligosaccharides, 5 to 15 grams per liter, which often exceeds the amount of total protein. Now, if we compare that to bovine milk, there's about 100 to 1,000-fold less oligosaccharides. The structures are very different. And since bovine milk is the basis for most infant formula, we hardly have any of these human milk oligosaccharides in infant formula, although that is starting to change. Now, a quick word about the structure of human milk oligosaccharides. What does that actually mean? Oligosaccharides comes from the Greek oligosakkar, a few sugars, and that's exactly what it is. It is a few sugar molecules, in fact, 5 monosaccharide building blocks that are put together in different linkages, in different combinations to make an array of different human milk oligosaccharides. Those 5 building blocks you see down here on the bottom are glucose, galactose, N-acetylglucosamine, fucose, and sialic acid. And depending on how we put these building blocks together, we get different structures, different functions. In the blue spot up here, I'm going to show you what we call the HMO blueprint. So that's the general formula, if you want, of human milk oligosaccharides. And then the white space around it will give a few examples of some of those oligosaccharides that you may hear of throughout this talk and others. So all human milk oligosaccharides start with lactose at the reducing end, and then they get extended by disaccharide units in different linkages. They can be branched, they can be further extended, and that just alone is a repertoire of backbone structures that we call the neutral non-fucosylated oligosaccharides. Now, if there's non-fucosylated oligosaccharides, you can imagine there is fucosylated oligosaccharides, meaning we can add fucose to these backbone structures, and we can do that in different linkages. If we add fucose to just lactose alone, we get the small oligosaccharides, 2-fucosyl lactose and 3-fucosyl lactose. If we add it to the growing chain and in different linkages, we get more complex structures that we also call fucosylated. Now, these would be our neutral fucosylated oligosaccharides. Well, if there's neutral, there must be something opposite of that. So there is the acidic or silated oligosaccharides, and that's because we can add sialic acid to the growing structures. Again, in different linkages, we can start off adding sialic acid to lactose alone, and we get 3 and 6-sial-lactose, and we can add it to the growing chain and just 1 sialic acid, as you can see down here at the bottom, and that would be our acidic or silated oligosaccharides. Now we can combine that and add fucose and sialic acid to the backbone, and that would be our acidic or silated fucosylated structures. So it's really a different way of putting these different monosaccharide building blocks together. Remarkably, there's about 150 to 200 different oligosaccharides that have been identified by structure so far. And really every mom makes a somewhat different fingerprint of what oligosaccharides are in her milk. You can see that here in data from the Canadian child cohort, where we see on the x-axis 1,200 different subjects, different milk samples, and on the y-axis the oligosaccharide concentrations in micromole per ml, different colors being different oligosaccharides. And it's evident how different that is between different moms. Low concentrations on the left, high concentrations on the right, the pattern being very different. And that is highlighted here again. If you take all those samples with a 100% abundance and now look at how the different oligosaccharides are distributed, it looks very different between different moms. So it's almost like a thumbprint. On the other hand, while we have huge inter-individual variation, we have very little intra-individual variation, so within the same subject, within the same mom, over a short period of time. So here you see milk oligosaccharide composition over the course of 7 days, and clearly there is hardly any difference. And if we go even higher in resolution and look at every hour, there is nearly a constant oligosaccharide concentrations here in the same subject. Again, very different between two different subjects. On the bottom now, a different subject, so the color pattern here looks very different for bottom and top, but looks remarkably constant over this short period of time. Over a longer period of time, you see that oligosaccharides, most oligosaccharides drop in concentration. So here we're talking not about several days, we're talking about from 1 month to 24 months, 2 years of age. And you see that many oligosaccharides drop in concentration with a few exceptions, one being 3-fucosyl-lactose up here and 3-sial-lactose down here. So there's a few oligosaccharides that go against the stream and increase over time, and we're trying to understand why that is. Now, here's a different representation of how oligosaccharide compositions can be remarkably different. This is data from about 10,000 milk samples, where every dot in this 3-dimensional space represents the HMO composition in the milk sample. And the closer those dots are together, the more similar the HMO composition, and the further apart, the more dissimilar. And you can clearly identify here that there is 2 different bubbles, if you want. So we call that HMO lactotypes, also known as secretors and non-secretors. And without going too much into detail what that means, it just means that there is different oligosaccharide compositions on the right than there is on the left. Both make oligosaccharides, but the oligosaccharide composition is very different. And these samples have been collected really from different parts of the world, but geography is not really what's driving this separation between secretors and non-secretors. Now, there is research focus on at least 3 different questions here. What are the maternal drivers that are responsible for the different HMO composition? Why is it different between different moms? And then, of course, what is the impact of these oligosaccharides on infant health and development, term and preterm? And last but not least, can we leverage some of that gained knowledge to develop new therapeutics for people of all ages, something we won't talk about in this presentation. Just one word or one slide really about the maternal drivers, since the focus should be on infant health and development for this presentation. We've seen that genetics, maternal genetics, are really the main drivers of HMO composition variation, to the point where this left and right distribution that I showed you earlier is driven by a single nucleotide polymorphism. So genetics play a major role in why we have different HMO composition profiles. But we've also seen lately that maternal nutrition, exercise, use of probiotics, maternal health, maternal drug use can all slightly impact human milk oligosaccharide composition. Why is that important to know? Because at one point, when we identify which oligosaccharides are good for certain infant or maternal health outcomes, we might be able to actually modify oligosaccharide composition by suggesting mom going on a different diet or using a different exercise regime or using a probiotic. So we're already setting the stage to potentially modulate oligosaccharide compositions if we find out which oligosaccharides are preferential. But now let's talk about infant health and development, which of course is the key interest. What do these oligosaccharides actually do? And what happens and how do we study that? So here's the research pipeline, as we call it, where we really use the entire spectrum from in silico models, in vitro and ex vivo cell models, and then go into animal models that are more or less suitable to study this. And then a very strong part is using human cohort studies, where we can analyze hundreds, thousands of milk samples for HMO composition, and then associate that with maternal drivers or infant outcomes. And last but not least, of course, we have clinical studies, and two of our speakers will specifically focus on that when it comes to HMOs and the current clinical studies. So all of that happens not only in one lab, it happens really in a network of collaborators from around the world. And since there's so many different topics where human milk oligosaccharides are currently under study, I want to focus on one specific aspect, and that is specifically on the very low birth weight infant, on the preterm infant in the NICU, and that is on necrotizing endocolitis. So necrotizing endocolitis is still a huge burden for the preterm infant. About 5% of all very low birth weight infants develop this often deadly disease. And on the other hand, we see that preterm infants who receive human milk are at a 6 to 10-fold lower risk to develop NEC. And we want to know what is in human milk that makes it so powerful to save preterm infants from this devastating disease. Now here in this space, I'm going to develop the long story of how we started to study human milk oligosaccharides in the context of NEC. So you'll see this slide come over and over and being filled in here, the white spaces. First, what we did is to address this research is to take an animal model, a rat model of necrotizing endocolitis, and ask the question, if we feed these rats with pooled human milk oligosaccharides, so all of the oligosaccharides isolated from human milk, do we get any effect on necrotizing endocolitis? And indeed we did. So if you look at the survival plots here, this is over the first 96 hours, you see that all the infants that stayed with the dam, all the pups that stayed with the dam, so the dam fed control here, all of the animals did not, none of the animals developed NEC. Whereas the formula fed without any oligosaccharides, we lost about 25% to NEC. Now, on the other hand, if we add human milk oligosaccharides to the same formula, you see that the survival goes up significantly and is almost to the level of the dam-fed control. So that to us was a huge positive surprise to see that oligosaccharides have such a positive effect here. We then went a little bit deeper, looked at the intestines, looked at histology of ileum sections here. We're able to score that with the help of a pathologist. And not only is survival different, but also you see that the pathology scores from healthy 0 to disease 4 is remarkably and significantly different if we add human milk oligosaccharides to infant formula. Now we wanted to know, well, which oligosaccharide is it? So we started to use a multidimensional chromatography approach to separate the oligosaccharides in different fractions and subfractions. So we didn't have to test all 150, 200 different oligosaccharides in, let's say, 20 animals per oligosaccharide, which was, of course, completely unfeasible. So we first separate oligosaccharides by charge. And as I said earlier, sialic acid introduces a negative charge. So now you have acidic oligosaccharides, and you can see that here, oligosaccharides separated by charge, -1, -2, 3, and 4. And the oligosaccharides that carried a negative charge 2, so 2 sialic acids, were the most effective in reducing pathology scores. And we then looked at this. There's more than one oligosaccharide, of course, that has 2 negative charges. And in the second dimension, we separated those now by size. So depending on how large the backbone is, we could separate that, and we then get a better resolution and find that there is one oligosaccharide here that is responsible for the protective effects that we see in this animal model. And we then used several different techniques here to identify how this oligosaccharide looks like, and we identified it as DSLNT, which stands for disialyl lacto-N tetraose. So it contains 2 sialic acids, disialyl, on this LNT backbone here. Now, that's great. That's an animal model. That is not a human preterm infant. And we, of course, now wanted to see, does this really translate to humans as well? So what we did next is we designed our own mother-infant pair cohort. Remember the power of cohort studies, where we specifically recruited moms and their preterm infants. collected milk samples that went into those infants about every second day, analyzed that for oligosaccharide composition, and then identified which oligosaccharides associate with a higher risk of NEC. And so briefly to go through this, you see that infants who develop NEC received milk with less of this protective oligosaccharide Dsnt than infants who do not develop NEC. The cohort was done in 5 different sites in North America. And here's an example how we matched cases and controls. Here on the top, you see a case. These are the first 28 days we had milk samples available every time you see a number in one of those boxes. These red boxes here highlights that the infant developed NEC during that time. So we had 4 samples before, 4 samples after NEC. And then we matched that based on gestational age, birth weight, delivery mode, when we have samples available, and so on, and then analyzed the oligosaccharides accordingly. So here's the results of that. If we looked at oligosaccharide total, so the sum of all oligosaccharides that we analyze over the first 28 days postpartum, we see there's no difference between controls, which is the dots, the gray dots here in the back, and the NEC cases, Bell stage 2 in yellow and Bell stage 3 in red. So there's no difference between controls and NEC cases for total oligosaccharides. But that wasn't the hypothesis to start with. The hypothesis was that there is this one oligosaccharide Dsnt that is potentially less in milk samples fed to infants that later develop NEC. And here is the data for Dsnt. It's exactly what we had hypothesized. Again, gray dots are the controls. Now the Bell stage 2 and 3 samples all cluster in the lower concentrations of Dsnt, which means there is less Dsnt, less of the protective oligosaccharide in the milk samples that was given to infants that later developed NEC. And here's a slightly different way to look at that. If we really look at each and every sample, color-coded now, so all these are our NEC cases. Red means the concentrations of Dsnt are lower than in the control, and all the samples here on the top for Dsnt show a red color, meaning Dsnt is lower than in the controls in every single sample. That's not the case for other oligosaccharides, so it's highly structure specific. And if we then model that and look at the odds ratios, you see that Dsnt really stands out as being significantly associated with a lower risk for NEC. So the more Dsnt, the lower risk for NEC, or the other way around. And we can then even go in and say, well, can we use that as threshold models where we say, well, if we take a milk sample and the concentration is higher than a certain threshold for Dsnt, then most of our infants will not develop NEC. If Dsnt levels are lower than that threshold, then we have to be careful, look at a few other parameters, and then make a decision whether we want to feed that milk to this infant or rather switch it or supplement. So this is great to screen mom's own milk, to say that your Dsnt levels are fine. You can give that to a preterm infant. We can also screen donor milk and human milk products like fortifiers to make a decision of whether we want to use that in the care of this patient. Now, these were 8 NEC cases in this study. We then collaborated with Dr. Chris Stewart, who's going to talk afterwards. And they have this wonderful biorepository of milk samples and many other samples from infants that develop NEC and have control samples for that. So we were able to analyze that. And here now you see we have 33 NEC cases, 7 controls. And there is clearly some difference here in the oligosaccharide composition. And if we narrow this down to which oligosaccharide is different, you can really see that there is one oligosaccharide standing out as being highly associated with NEC, and that is, again, Dsnt. So the more Dsnt, the less likely to have NEC. The less Dsnt, the more likely to have NEC. And Chris will talk about that in the next talk. So filling in the blanks of this cartoon here, so Dsnt not only was found to be protective in the animal model, it was also found to be associated with NEC risk, less Dsnt, less of the protection, meaning higher risk for NEC. So that now gives us greater confidence to move into clinical studies, and we're going to talk about that in the future talks as well, and also to study the mechanisms of how this actually works. Now, to connect those two different things, we've shown that HMOs at least have some part in this protective mechanism of human milk again. And not only HMOs in general, but really Dsnt, one specific oligosaccharide in particular. We've seen that a specific HMO is associated with lower neck risk, and the same HMO improves survival and neck pathology in an animal model. This is, of course, a great opportunity to develop new therapeutics based on Dsnt, and it's also an opportunity to develop new diagnostics to screen mom's own milk and donor milk products. So this is NEC. This is one example for the preterm, very low birth weight infant. Oligosaccharides have been associated with many, many other things as well, mostly in the term infant space. For example, when it comes to diarrhea, pathogen exposure, and some examples for 2FL, Campylobacter, enteropathogenic E. coli, so potentially some protective mechanism. LNT has been associated with group B streptococcus infections, but also with protozoan parasites like Entamoeba histolytica. We've seen that other oligosaccharides are associated with rotavirus infections, and you can go on and go through different pathogen types, whether it's viruses, bacteria, protozoan parasites, and different oligosaccharides having potential protection. We also see there's oligosaccharides being studied in infant growth, and 2FL again pops up being associated with growth in infancy that extends to early childhood. Neo-LNT plays a role here because the ratio between those two is important. And we now have 3 or 4 different human milk cohorts or human cohorts where we show the same associations over and over and over again. So it gives us greater confidence that there is something real that's not just a bystander effect association. And then on the other side of the spectrum of growth and malnutrition, we find that the silated oligosaccharides seem to be effective here. One more example, neurodevelopment and cognition. We see that 2FL here again stands out being associated with memory and learning in animal models, but we've seen this recently in a human cohort as well, that there's associations between 2FL concentrations at 1 month and 24-month cognitive development in the infant. And then the silated oligosaccharides play a role here as well. And one more for immune development, different oligosaccharides being associated with certain cytokines that being released with chronic macrophage inflammation, which we study extensively. And there's a review that goes into this in detail and links different oligosaccharides with different immune responses. Very important here is to see that in the cohorts that we've done, we very often see that it's not an individual oligosaccharide that is associated with outcome when it comes to the immune response, but it's really a mix of different oligosaccharides, either being here associated with cow milk allergy, with food sensitization, or with allergic disease, short-term and long-term. And here we looked at all the way out to 18 years of age. So it seems to be of a mix of oligosaccharides being responsible. So a couple more words of how this all works. What is actually the mechanism? How do these oligosaccharides do that? And we strongly believe that oligosaccharides are more than just food for bugs. And I'll explain that in a second. But before we get to that, one word about what happens to the oligosaccharides actually when they get ingested. They're very resistant to the low pH in the stomach, also are not digested by pancreatic or brush border enzymes. 1% is absorbed, makes it into the systemic circulation, and is excreted intact with the urine. And then the rest is either degraded in the colon by bacteria or is excreted intact, and we find that in the diaper. And in this cartoon, you see really the interplay between oligosaccharides here on the top, good bacteria here in green, potential pathogens in purple, the intestinal epithelial cells on the bottom, and then underneath that, immune cells that are either associated with the intestine or act on a systemic level, since oligosaccharides can make it all the way through. So we see that oligosaccharides are prebiotics, so they are degraded, they're utilized by certain bacteria. Those bacteria have a growth advantage and potentially have a benefit on the host, on the baby. But we also see that oligosaccharides specifically go after certain pathogens. Epithelial cells, they directly affect immune cells, and all that can then feed back into the intestinal lumen here. And once again, there is local effects, but oligosaccharides make it into the systemic circulation, and there can be effects on organs, on tissues, other than those in the gut. So that's something to keep in mind. And last but not least, we clearly think that we are only at the very surface here, at the tip of the iceberg, and there's so much to discover what all these oligosaccharides can do. All right, so to summarize this introduction to oligosaccharides, HMOs are the third most abundant solid component of human milk. It's a group of complex sugars, complex glycans, and there's about 150 different ones. Some are small, like 2FL, 3FL, 3SL, and 6SL, or they're larger, like very strongly branched structures, high molecular weight structures. Then we have some that are either acidic, so they contain sialic acid, or they're neutral, no sialic acid. And then they can be fucosylated or not fucosylated. The structure determines HMO function, and this is important. Not all oligosaccharides do the same thing. There's a reason why we have 150 different ones. They're not digested by the host, by the baby. There's local effects in the gut, as well as systemic effects after absorption. They're serving as prebiotics, but they're doing so much more than just being food for bugs. They're anti-adhesive, antimicrobials, epithelial cell modulators, immune cell modulators, and a lot more. And that way they can affect outcomes like infectious diseases, diarrhea, growth, neurodevelopment and cognition, immune system development and response, and as I've shown you in the example here, particular necrotizing endocolitis, but we also have some data on sepsis. And once again, this is probably just the very surface, and there's so much more to discover. Now, we see that HMOs contribute to shaping the infant gut microbiome, which may impact infant growth and development. And you will hear more about that from Dr. Stewart in the following talk. HMOs are prebiotics, but also more than just food for bugs. So really, there is an opportunity to find the right balance of what oligosaccharides should be utilized when we develop prebiotics, probiotics, and symbiotics. Sometimes you really want to spare a certain oligosaccharide like Dsnt that is not utilized by your probiotic of choice. So it's still available to do other things. We need a combination of preclinical, cohort, and clinical studies to fully assess and validate the effective functions of HMOs. And Dr. Sayevska will show you in this session what kind of studies have already been done in the term infant. And then in the following, Dr. Hescott will talk about specifically the preterm infant. And last but not least, human milk is not just HMOs, right? I mean, it's my favorite topic, but I know there is so many other bioactives in human milk, and it's really a part of dynamic co-adapting mother milk infant triad, as we call it. And Dr. Cavallo will talk about that at the very end and give us a little bit of a future perspective bioactives there are and how we tackle this really complex system of this triad. So I leave you with that and go to the next speaker where we talk about more about the microbiome and what oligosaccharides can do there. Thank you.